Royalactin in queen development, molecular mechanisms of caste determination in Apis mellifera by Eva Zane. Caste differentiation in honeybees is driven by nutrition with genetically identical larvae developing into either workers or queens. A key factor is royal jelly, particularly the protein royalactin, which activates pathways for growth and reproduction, leading to the queen phenotype. Studies on royalactin provide insights into colony health, epigenetics, and developmental flexibility. Royalactin is a glycoprotein found in royal jelly and is considered essential for the development of queen bees in Apis mellifera. It belongs to the MRGP1 protein family and undergoes secretion and post-translational changes. Its effects are thought to be mediated through activation of the epidermal growth factor receptor, EGFR pathway, which supports larval growth, cell division, and differentiation. The honeybee Apis mellifera displays phenotypic plasticity, where the genetically identical female larvae develop into either fertile queens or sterile workers based on their diet. This is primarily due to royal jelly, a substance rich in proteins, lipids, vitamins, and other bioactives, which is fed exclusively to future queens. Royalactin, a key component, activates EGFR signaling to promote queen traits like ovary development and larger size. However, research suggests that other royal jelly components, such as fatty acids, microRNAs, and MRGPs, may also play critical roles. My research question is, what components of royal jelly are important for the queen bee development? My first piece of data is a figure that shows the survival of enclosed honeybees provided with various fractions of royal jelly. So as we can see, we have 5 different fractions of royal jelly and 1 of pure sucrose. And the one that has the longest survival rate and is most successful is the royal jelly 4%. All these different fractions have different concentrations of royal jelly and sugar or sucrose. But as we can see, the pure sucrose concentration is coming in last. It has the shortest lifespan, the lowest shot at life. So this just goes to show that royalactin is an essential and vital part of queen bee growth and development, because without it, the lifespan would be more than cut in half as if they were to have it. The second figure shows diet, quantity, and principal component relationship. This shows that caste determination is very heavily linked to the diet. The increased diet quantity of the royal jelly leads to increased queen traits, meaning more of the diet that is consumed, the better the outcome. So this definitely has a very strong link to quantity, diet, and the successfulness of queen bees and how they express their traits. Figure 3 shows increased juvenile hormone promoting queen bee growth in Apis mellifera. So as we can see here, the queen bee has surpassed the threshold twice, the worker falling very short. So the royal jelly, extended and abundant royal jelly feedings allows for the juvenile hormone to be more expressed, and then that will trigger the queen-specific growth. So they're all kind of linked to one another, but the more royal jelly, the more hormone, the greater the expression of queen traits will be. So again, that is very heavily linked. As we can see here, the queen bees have surpassed the threshold, and it's not even comparable to the worker bees due to the juvenile hormone that is being consumed and given to the bees. In summary, we can see that queen development in honeybees is deeply influenced by both the quality and quantity of royal jelly. Key components like royalactin regulate gene expression and hormone levels, shaping the bee's development. Research shows that nutrition plays a vital role not only in determining caste, but also in the overall health and sustainability of the colony. These findings emphasize the importance of proper environmental and nutritional management for successful queen rearing and colony growth. Excuse me, I'm sorry. Okay, and here we have our significance. There's quite a few significant findings for this research, kind of just demonstrating exactly how important royalactin and royal jelly is to queen bee development. Understanding the molecular mechanisms behind honeybee caste recombination, especially the role of Royalactin in larval nutrition, can definitely improve queen rearing practices. These insights can lead to healthier, longer lived queens, helping combat colony loss and support sustainable beekeeping, biodiversity, and global food security. Um, this just covers a little bit of how important it is. It is so essential to queen bee development. Without Royalactin, larvae develop into sterile worker bees instead of fertile queens. It's essential in determining the caste differentiation within the hive, and the honeybees couldn't function without this kind of caste differentiation that Royalactin is essentially in charge of. It triggers a unique and genetic physiological response in bee larvae, promoting faster growth, increased body size, and development of functional ovaries. Here we have all of my references that I use in the introduction, background, and 3 figures. Hope you enjoyed my presentation. Thank you.